All right. I have the pleasure of introducing Alison Betts of Takeda. Alison has 31 years of experience in DMPK, PKPD, and QSP modeling and simulation. She began her career at Pfizer UK in 1994 and worked across 3 sites for 25 years, notably supporting oncology research and drug approvals. In 2019, Alison became VP of Scientific Collaborations at Applied Biomath, receiving an NIH SBIR grant. In 2023, she joined Takeda as Global Head of DMPK and Modeling, leading scientists across Boston and Japan. Alison holds a PhD from the University of Leiden and a BSc from the University of St. Andrews, with 31 publications and 40 invited presentations. And she's going to talk about quantitative systems pharmacology platforms to predict efficacy and toxicity for ADCs. Alice.
Thank you so much. I'm very happy to be here to talk about our steps towards QSP, Quantitative Systems Pharmacology, platform model to predict ADC efficacy and toxicity. Okay. So I obviously don't need to explain to this audience that ADCs are an extremely exciting modality, which are clinically validated for the treatment of cancer. So there's over 13 approved ADCs now, targeting various solid and hematological malignancies. They have payloads which have multiple mechanisms of action. So you've heard about the tubulin inhibitors today. There's also the DNA damaging agents and the topo-1 inhibitors. They have transformative responses in the clinic, so they're both broad and deep, even in late lines. And there's several ADCs with accelerated approval. And also the checkpoint inhibitors have emerged as obvious and validated combination partners. However, this has taken us a long time to get to this stage. There's been a significant journey. There's been a number of clinical failures for ADCs. And I think this quote comes from Greg Thurber, so I think I'm quoting you here when I say that ADC concept may be simple, but the application is difficult. And this is really because ADCs are multivariate molecules. So the optimal properties of an ADC depend not only on the antibody, the linker, and the payload, but they also depend upon the properties of the tumor target, the indication, and the patient. And the design properties can be really quite counterintuitive. As you saw today, talking about Trodelvy, for example, Peter Center talked about Trodelvy, which has very different properties than what you'd expect for a successful ADC. The translation from preclinical studies is also complex, and I'll talk about that more today. And most importantly, they do have a narrow therapeutic index, so they're not truly targeted molecules. And when I first met Professor Wittrup, which was, I think, about 12 years ago, then we'd been talking in the industry about these being site-directed missiles. So the story was that they go, straight to the tumor, release the payload, and then kill the tumor cells. But Professor Wittrup described these as being more like tethered mines. And he's completely right. I mean, they go all around the body. You do get uptake of ADC and release of payload in normal cells. But in general, for ADCs, you get more in the tumor. And that's what's driving their success, their efficacy. So what I'm going to talk to you about today is quantitative systems pharmacology modeling. So I know that we've got a mixed audience here. So I want to tell you a little bit about what QSP modeling is. So basically, if you show a picture of the ADC mechanism of action, then quantitative systems pharmacology modeling basically just turns that into math. It basically turns all of the reactions, the interactions into a series of ordinary differential equations. And then we can inform that model using data. So this can be data from the literature, it can be in vitro data, it can be system properties like receptor expression and internalization. And we can also use the model and fit it to data sets. And I'll show you some of this in my talk. And then the final model that you have, you can use it for simulations. And this can be to answer questions like, what kind of dose do I need in the clinic? How do I design my clinical study? Or earlier on, it can be, how do I design the optimal ADC? You know, what kind of affinity do I need for this kind of a target? And so I think that QSP modeling is a useful, almost essential tool to guide ADC design and development in the face of complexity. Okay, so today I am going to talk to you about a next generation platform systems pharmacology model that we developed for preclinical to clinical translation of ADC efficacy, and how we used it for dose predictions, for including outcomes, so for predicting RECIST criteria and differentiation from standard of care in different oncology indications. I'm also going to talk about a bystander effect model that we've used for more design-based questions, for optimizing drug properties and predicting therapeutic index. And then finally, I'll talk about a mechanistic model for predicting ADC-induced hematological toxicities, and today I'm going to present on thrombocytopenia. So all of this work was funded by an NIH grant, and this was a grant that I applied for when I was working for Applied Biomath, which has now become Certara, and this was direct to phase 2 small business innovation research grant that was funded in 2020. Because what else do you do in a global pandemic? But okay, so talk about the efficacy model first. Okay, so this is a picture of the model schematic, and this work builds upon previous models that have been developed by myself and Deval Shah and then others in the industry. And it's basically a series of sub-models, and these are linked together in a modular fashion. So the core of the model, quite literally, is the tumor cell model. And this is also the in vitro model. And this incorporates target positive and bystander cells. Importantly, it captures the whole mechanism of action of the ADCs, including binding to receptor on the tumor cell surface. It also accounts for release of payload in the tumor environment. So you've got extracellular release of payload and also competition with naked antibody for binding to the receptor. The model accounts for internalization and also recycling of the receptor, and it incorporates the intracellular mechanism of action of different linker cleavage mechanisms. So we talked about the difference between the non-cleavable and the cleavable mechanisms, and that's included. It includes the release of payload, transport out of the lysosomes or diffusion, and then binding to intracellular targets. And then it also accounts for diffusion of the payload out of the cells or active efflux. So this is like the real epicenter of the model. And then basically what you've got attached to it is kind of a fancy PK model. So this model really describes the disposition of the ADC, the antibody, and the payload in 3 compartments. So the central compartment, or the blood, peripheral, which is all of the tissues, and then in the tumor, of course. And we include some separate sub-modules. So we've got, for example, the module to describe binding to soluble target if you've got shed target. And we also include binding of ADC to healthy cells expressing the tumor target. And so really the ADCs have a very complex disposition, and the model kind of tries to incorporate most of it. We also have a tumor disposition model, which is based on a cross-cylinder model, which I won't go into today, but I'm sure Greg's going to talk about this more in his presentation. So the model predicts the intracellular payload concentrations, and then this is linked to a mechanistic tumor penetration model, which is based on the Simeone model. It's linked to a tumor growth inhibition model. And then we also have a semi-mechanistic and a mechanistic hematological tox model. Okay, so first of all, the platform model was developed initially using 2 HER2 targeted ADCs, which luckily you've heard a lot about today. So this was basically Kadcyla or TDM1 and Enhertu or TDXD. And the reason that we picked these 2 ADCs for model development was, first of all, because there's a lot of data in the public domain on both of those ADCs for model calibration, so that was great. And also they're very interesting because while they both have the same antibody, they both have trastuzumab, and they both bind to HER2, they do have different mechanisms of action. So they have different payloads, they have different linkers, and they also have a different DAR. And personally, I was always also interested in seeing if we could use the model to figure out, what makes these so different and what makes TDXD such an interesting drug. So subsequently, the platform model has been applied to other ADCs, including Tivdak and Trodelvy, applied biomass, and also ADCs which have failed in clinical development, because these are probably more interesting. So key data that is in the model includes data from a number of different sources. So first of all, there is physiological parameters. So the PK model is a physiologically based model. Then there are cellular measures, so data like receptor expression, internalization rate, tumor growth rates, which are mainly fixed in the model. And then there's the drug properties, so things like the drug to antibody ratio, or the DAR, the drug affinity for its tumor target, and the payload efflux. And then there's data that we use to actually calibrate and validate the model. So that includes in vitro cellular disposition, pharmacokinetics, tumor growth inhibition, thrombocytopenia data, and also outcome data in the clinic. So first of all, I want to talk about the calibrating the in vitro cell model, so like the core of the model. And for HER2, there was a lot of data published on trastuzumab and its internalization and recycling, which was great because we were able to use that data to really get that component of the model right. And then there was this really useful data. And I always tell people that if you want to do one kind of experiment with ADCs, do this in vitro kind of disposition experiment, because it gives you a lot of information. So what the authors had done here was that they'd taken TDM1 and they'd incubated it with different cancer cells, and then they had basically figured out the disposition of the ADC and the payload. So they measured total DM1, conjugated DM1, and unconjugated DM1 inside cells. They had measured unconjugated DM1 in the media, and then they had measured total unconjugated DM1 in the media and the cells. So this gave us a lot of information to fit the model to the data, and really from then on in, we could fix all of this, the in vitro model to the parameters that were determined in this model. And there were some interesting observations. So the intracellular DM1 concentrations are much higher in the tumor than intracellular than in the media, which obviously makes sense, but this is really how ADCs work. Okay. So now that we had the tumor model, the next part was to calibrate the model to mouse data. So we basically took the intracellular, the tumor model here, and then we added a PK model. Now the PK model was kind of simple for TDM1 and TDXD in mouse, and that's because trastuzumab doesn't bind to the rodent form of HER2, so you don't have to account for soluble HER2 or for binding to normal cells. So we basically put on the simplified PK model, attached it to the tumor model, and then we could predict the payload concentrations inside the tumor cell. And then the tumor payload concentrations were linked to this model of tumor growth and tumor cell killing. So then what we could do was we could use the mouse model to simulate the tumor payload concentration and then link it to tumor growth inhibition. So we had quite a lot of data sets that we took from the literature where we had a lot of TGI data. One thing I should say, there was a lot less data on TDXD available than TDM1, but we had enough to bridge because they're very similar in their mechanisms. So for example, we had concentrations of TDM1 in the central, like the blood, in the tumor, and so we could check that the model was kind of behaving itself. And then, so all of this was simulated up front without having to estimate any parameters. And then the TGI data we actually fitted to the tumor growth inhibition data in the mouse, and that's where we used the model to estimate the tumor cell killing using the model. Okay. So the next step then was to calibrate the model to TDM1 and TDXD clinical PK data. So if you were using this model in a priori simulation mode, then you would predict the PK from preclinical data, and that's actually relatively straightforward to do. Now we've learned a lot about ADCs, but we had the PK data, so we used it and we fitted the model to it. But all of the lines here are the simulations, and they are actually not fitted to the clinical PK data. This is all using the previous parameters and actually just simulating the data. As you can see, the PK model is a lot more complicated for the human model because we had to account for binding to the normal tissue HER2 and also binding to soluble target. Okay. So now that we had the model set up, then we could use it in simulation mode to simulate outcome. So what we did was we parameterized the model. We actually parameterized for both HER2 positive breast cancer and also for gastric cancer. I'm just showing you the breast cancer here because that's what we published. So first of all, we included HER2 expression levels in HER2 3+, 2+, and 1+ patients. And because I'm a quantitative person and for the model, then we had to parameterize this as number of receptors per cell. And so these are the values that were put in. We also use clinically plausible initial tumor volumes for metastatic breast cancer and maximal tumor volumes, and clinically observed human growth parameters for HER2 positive MBC. The clinical PK parameters were taken from the PK model that I showed you in the previous slide. And the drug effect parameters were all estimated from the mouse. So from all of the mouse analysis that we had done, including the kill rates and the potency values, we used directly from the mouse. So these are some of the simulations that we did. So on the left-hand panel, you've got predictions of the TDM1 and TDXD PK, including the payload concentrations in the tumor and also the efficacy. And because it was a model, we could do like a head-to-head trial with the same doses. So we were able to simulate high doses of TDM1, similar to the high doses of TDXD that are seen in the clinic. And what we observed was that the tumor DXD concentrations were much higher than the DM1 concentrations. Now, some papers say that they're equivalent in potency. You know, you can debate that. But yeah, because you're getting more concentrations of payload in the tumor, then this is likely to be driving the greater predicted efficacy, or one of the drivers, I should say, of the greater predicted efficacy. We then used the model to do clinical trial simulations. So we basically simulated for 100 patients, and we followed the same. So we basically took the clinical trials that were run for TDM1 in phase 1 and phase 2, and TDXD, though not all of the trials that were presented by the previous speaker, but we took the phase 1 and phase 2 trials for metastatic breast cancer. And in the model, we also stratified between high HER2 expression and low HER2 expression, as had been done in the clinical trials. So the lines here are the model predictions, and the dots are the simulations. So you can see that the predictions are closely aligned with the observed data, and that following administration of TDM1, you get greater PFS in patients with higher HER2 expression, and with TDXD, PFS was greater overall and was similar between high and low HER2 positive patients. Okay. So next, I just want to talk a little bit about the bystander effect model. So I'm not going to go into this in a lot of details, but just to let you know that we took the Enhertu model, we expanded it to predict the uptake of DXD into bystander cells, not expressing target, and used it to predict intracellular concentration of DXD in HER2 positive healthy cells. And the reason that we did that was we wanted to try and get to the prediction of the therapeutic index. So because the TI is obviously a big design factor for ADCs, so what we did was we calculated the TI as the payload bound to topo1 in tumor cells over the payload bound to topo1 in healthy HER2 plus cells. And this is kind of shown on this plot here, where you can see that the tumor concentrations are much higher in the yellow, or the payload bound to topo1, compared to the central and the peripheral concentrations. And so we estimated the TI to be between 6 and 15, depending on the methodology that we used. So this kind of gives a way of predicting TI up front. Okay, so in the last 5 minutes, I just wanted to talk to you about the ADC-induced toxicity model. So, as you know, a major reason for ADC failures in the clinic is the inability to reach the efficacious dose without the onset of side effects. And these are usually severe hematological toxicities, and this is a major reason for drug holidays. And this table here shows a number of different ADC payloads, and all of the different classes have hematological toxicities. So we thought we'd look into developing a more mechanistic model for neutropenia and thrombocytopenia. I'm just going to show you the thrombocytopenia one today. So what we did was we basically took the whole stem cell to platelet differentiation, and then we parameterized it. So we enabled the stem cells, progenitors, and megakaryocytes to expand or differentiate, and then they produce megakaryocytes. So megakaryocytes produce hundreds of platelets per day, and then they die. And we included the thrombopoietin mediated feedback mechanism in, because this is really important in maintaining the homeostasis of platelets. And then what we did was we added the drug effects. We had the concentration of ADC driving either the death of megakaryocytes or like the reduction in differentiation. And here it is important to note that the ADC concentration was used as a surrogate for the intracellular payload concentration. So we're very lucky to have this data that was published on TDM1. So this is TDM1 given at the same dose, its maximum tolerated dose of 3.6 mg per kg, given Q3 weekly to 15 patients. So we actually had to digitize this data in order to calibrate to the model. It's so variable, we actually only managed to digitize 10 out of the 15 patients. But you know, it was a real gift to have this data. It's very unusual to get it. So then what we did was that we fitted the model to the 10 patients that we could digitize. First of all, as I said, there was significant variability between the patients, including starting platelet number, reduction in platelet count, and also in the TPO-mediated feedback response, which causes a big rebound in the platelet concentrations. And we could do this by calibrating the model to only 3 parameters: the platelet residence time, the generation rate, and the TPO half-life. And what we found was that the model could predict TCP grade for 9 out of 10 patients. So this was kind of like a good start. However, what we then went on to do was to really try and capture the payload uptake into CFU MK, because this was the real driver for the toxicity. It's not the ADC concentration, it's really the intracellular payload concentration. And the uptake into normal cells is driven by both target-independent mechanisms, so macropinocytosis, and also target-dependent mechanisms, so Fc gamma receptor binding. So I don't have time to go into a lot of the details on this, but this model is underway at Applied Biomath, and I'm sure they will be talking about it at future conferences. So I just want to wind up and say thank you to the members of the grant team at Applied Biomath, and I'll stop there and ask for questions. Thank you.
Thank you for that excellent talk. Really important work here. I am wondering if you can help me understand between the different compartments where the payload is at different times in the therapy. So on day 0, what is the breakdown of the payload within the intracellular compartment, within the cancer cell, within the tumor stroma, the ECM, and the intravascular space? And on day 21, what does that look like?
Wow, that's a really difficult question. Do you mean in patients?
In patients, or however your model predicts.
Well, first of all, it does take a long time for the ADC to distribute into the tumor cells. Mm-hmm. So you're looking at at least a day to get there. Right, the next After that, it's much faster. So you do get fast, especially with HER2, because it's like kind of rapidly internalizing. But to give you all the I'd have to go into the model with you, but we can have a look at it if you want afterwards.
That'd be great, thank you. Thank you.
Great talk, Alison. I just want to make sure I understand, because Gerald showed the data that TDXD gave better clinical results than TDM1. And this has led to some sort of conventional wisdom in the field that topoisomerase inhibitors are a better ADC ligand than antimicrotubule drugs. But if I understand what you're saying, that is not necessarily true, and it had more to do with the pharmacokinetic and pharmacodynamic parameters of the antibody drug conjugate than the efficacy of the actual drug. Is that correct?
Absolutely.
Okay.
So there's no reason why, topo 1s are kind of like the flavor of the month, and they work very well combined with certain targets. But I really sort of see this as a disposition problem. So that's why you've got to look very carefully at what the receptor expression is, what all the internalization rate is, the recycling, all of the different parts. So it can be that very, very potent payloads, like the DNA damaging agents can work really well. It depends upon the other properties.
Thank you. What I believe is that only 0.2 to 0.4% of drug actually go to target. Is this model can predict the 0.2 to 0.4% of ADC go to target, to be more specific? Is this model can predict where other drugs, other molecules, where the other molecules of ADC goes? Is that?
I can't hear
what
you.
Percentage of the yeah, so only 0.2 to 0.4% of the ADC actually go to the target. That's what I understand from most of the clinical trial data. So is this possible to predict this model, that what percentage of this ADC actually go to non-target tissue?
Yeah, because it's quantitative, so you can definitely predict how much gets there. It's a tricky question, because generally about 20% of ADCs actually make it into the tumor. Do they make it into all of the tumor cells is a completely different question. So because you have the binding site barrier, you can have problems with the ADC actually diffusing into the tumor, and that's why you end up with necrotic cores. So That's probably Greg's talk, I think. I'm setting him up.
Thank you.
You mentioned that you had also modeled gastric cancer in addition to breast cancer. I was wondering if you could describe the differences in those models and if you were able to recapitulate some of the differences in efficacy that have been seen in clinical studies in HER2 ultra-low in gastric cancer versus breast cancer.
Yes, so that's a great question. Yes, we can. So, but the assumption that's gone into the gastric model, I guess it's not really assumption, but from the literature, gastric is a lot more heterogeneous in terms of its HER2 expression. So the difference is in that in metastatic breast cancer, you've got a high number of cells which express HER2, whereas in gastric it's much lower. So with that in the model, then we can predict the fact that TDXD will work and TDM1 won't, using virtual patient simulations.
I was just wondering, maybe we can take this offline afterwards, but the differences in ultra-low in just in TDXD in gastric versus breast cancer.
No, we haven't done that actually. Oh. What the simulations that we did with TDXD were all assuming fairly high HER2 expression. I mean, again, this came from the literature because that was our first hypothesis, if you like, that the difference was mainly in the heterogeneity. So we didn't include like really low HER2.